Associative processes. Background. Like many animals, Japanese quail display stereotypical behaviors during courting. Males puff up their feathers, stretch their necks while they strut around, and emit a vibrating vocalization that lasts for several seconds. In some cases, a male will hold a worm in his beak to entice a female closer. When she approaches, he mounts and attempts to copulate with her. Males exhibit these courtship behaviors when females are present, but also when they are exposed to stimuli previously associated with receptive females. Domjan and his colleagues have studied these conditioned mating behaviors in great detail, showing that male quail prefer females that are the same color as those that they copulated with in the past, and that they are quicker to mount females if they first encounter stimuli that predict her arrival. These cues may also affect physiological responses. Testosterone levels are elevated, and more sperm is released during copulation in the presence of sex-related stimuli. In writing about his own work, Domjan 2002 notes that it is not surprising that sexual behavior is influenced by so many different factors. Without it, no species would survive, so it makes sense that the behavior is flexible enough to be elicited by more than one set of stimuli. The importance of his work is in showing how sexual responses are altered by experience and how this experience can have a positive effect on reproduction. In other words, forming an association between environmental stimuli and sexual experience facilitates short-term behaviors, i.e., courting and copulation, but also increases long-term consequences of these behaviors, i.e., reproductive success. Associative learning may also influence sexual responses in humans, although most of the work on this topic relates to deviant behaviors. Domjan's work builds on a long philosophical tradition. The idea that learned associations affect behavior dates back to Aristotle, 384 to 322 BCE, who formulated a set of principles describing how ideas or thoughts are connected. These were expanded in the 17th and 18th century, the 1600s and 1700s, by the British associationists, expounding the view that experiences are held together by associative connections. Uncovering the properties of these associations would explain how knowledge is acquired. As noted in the previous chapter, Ebbinghaus tested the laws put forth by the British associationists, using himself as a subject. Based on these findings, he presented a series of associative laws explaining how memories are formed. Ebbinghaus, 1885. With the turn of the 20th century, the turn of the 1900s, and the rise of behaviorism, the study of associative mechanisms took hold in North America. Many scientists were also influenced by the pioneering studies of Ivan Pavlov, 1849 to 1936, a Russian physiologist who won a Nobel Prize for his work on the digestive system of dogs, but is far better known today for his work on conditioning, a form of associative learning. Chapter plan. This chapter begins where Pavlov's work took off, summarizing the last century of scientific research on associative processes. The chapter focuses on classical and operant conditioning, which are the two most commonly studied forms of associative learning. In order to understand how research in this area is conducted, the primary experimental paradigms for operant and classical conditioning are described. This is followed by a discussion of how an animal's evolutionary history impacts its ability to form associations. The mechanisms of classical and operant conditioning, as well as the phenomenon of extinction, are outlined as a means to understand how associative learning operates. This leads to an overview of the theories of associative learning. Finally, as with all cognitive processes, associative learning is represented by changes in the central nervous system. There is extensive evidence pointing to specific brain systems that control both classical and operant conditioning. Although the evidence is not complete, these provide working models for future investigations of associative mechanisms. The majority of research on associative processes has been conducted by experimental psychologists working in the lab, although there are compelling and important studies of both classical and operant conditioning in the natural environment. These tightly controlled lab experiments allowed researchers to identify mechanisms of associative processes, including which environmental conditions produce the strongest conditioning. Ethologists and behavioral ecologists studied some of the same phenomena as a means to understand the evolution of naturalistic behaviors, although they seldom discussed their findings in terms of associative processes. By the 1960s, there was a growing realization that these two approaches are complementary. Today, many researchers consider both proximate and ultimate explanations of associative learning, even if they continue to work only in one tradition. Terminology. Every organism lives in an environment surrounded by hundreds or even thousands of stimuli. Those with motivational significance will elicit a behavioral response or, more accurately, a combination of responses. Classical conditioning. Stimuli that do not initially elicit a response, at least not one that can be measured, may acquire the ability to do so through association with motivationally significant events, a process known as classical conditioning. The systematic study of classical conditioning began with Pavlov's work on conditioned reflexes. Indeed, the two are so closely connected that the terms classical and Pavlovian conditioning are used interchangeably. In the simplest version of Pavlov's experiments, a researcher rings a bell and then presents a dog with food, see figure 4.1. This sequence is repeated and, eventually, the dog salivates when it hears the bell, presumably in anticipation of the food. Pavlov identified 4 components in these experiments that are now common terminology in classical conditioning: the unconditioned stimulus, US, food; the unconditioned response, UR, salivation; the conditioned stimulus, CS, bell; and the conditioned response, CR, salivation. Prior to conditioning, the unconditioned stimulus elicits the unconditioned response. This is sometimes called the unconditioned reflex. Following conditioning trials in which the conditioned stimulus is paired with the unconditioned stimulus, the conditioned response is elicited by the conditioned stimulus, sometimes called a conditioned reflex. In the case of Pavlov's experiments, a dog salivating to the sound of a bell would be a conditioned reflex. Observation of this very simple response was the basis for an entirely new discipline. See box 4.1. Figure 4.1. Ivan Pavlov and his fellow lab mates from the physiology department at the Military Medical Academy in Petrograd, 1914. Box 4.1. Psychoneuroimmunology. As with many scientific breakthroughs, the demonstration that immune responses can be classically conditioned was a chance discovery. In the early 1970s, Adder and Cohen, 1975, were working with the immunosuppressant cyclophosphamide. The drug is used to inhibit natural immune responses in a wide range of clinical conditions, organ transplants, autoimmune disorders, but it produces terrible side effects, including nausea. Adder wanted to find a solution to this problem, so he allowed mice to drink a flavored saccharin solution prior to an injection of cyclophosphamide. As expected, the mice developed an aversion to the taste of the solution. In order to test the effects of various anti-nausea agents, Adder force-fed the saccharin solution to these mice. Unexpectedly, the animals kept dying. At the time, the explanation was not obvious, but it is now clear that the saccharin solution was suppressing immune responses in animals that had experienced the saccharin cyclophosphamide association. Adder demonstrated this experimentally by showing that mice exposed to the flavored saccharin water, i.e. the conditioned stimulus, prior to an injection of foreign cells, developed fewer antibodies or a weaker immune response than did mice exposed to water. Subsequent work showed that classical conditioning can also increase immune system activity. In these experiments, an odor was paired with an injection of the drug interferon. Interferon increases the activity of natural killer cells in the bloodstream, which help the body to fight off infections, viruses, and foreign cells. In both mice and humans, an odor-conditioned response paired with an interferon injection increased natural killer cell activity, even in the absence of the drug. From a rather serendipitous finding that classical conditioning affects immune responses, an entirely new discipline emerged. Psychoneuroimmunology examines the interaction between psychological processes, the nervous system, and immune responses. Another way to think about this is how the immune system relates to brain function and how these alter cognition and behavior. An obvious application of this work is the study of immune system disorders and how these impact mental health. Operant conditioning. In classical conditioning, an unconditioned stimulus is presented following the conditioned stimulus, regardless of how the animal responds to the conditioned stimulus. Pavlov's dog did not need to do anything in order to receive the food after a bell rang. In contrast, an outcome, such as food, is presented in operant conditioning only if the animal performs a response. In that sense, the definition of operant conditioning is a change in behavior that occurs because the behavior produces some consequence, positive or negative. The prototypical example of operant conditioning is a rat pressing a lever to receive a sugar pellet, but any behavior that produces an outcome, either in the lab or the natural environment, can be described as an operant. This includes a cat pulling a rope to escape from a puzzle box, a rodent swimming to a hidden platform in a water maze, a bird pecking at tree bark to obtain food, or an animal avoiding a location where a predator was encountered in the past. Thorndike, who pioneered the study of operant conditioning in the lab, noted that animals tend to repeat behaviors that produce satisfying effects and refrain from repeating those that lead to unsatisfying effects, the law of effect. A premise of this idea is that behavior is controlled by its consequences. This relationship between a behavior and its consequences can be expressed in one of four ways. See Figure 4.2. 1. Positive reinforcement describes a contingency between a response and an outcome that increases the probability of the response occurring. Giving a dog a treat when it comes to a call, praising children for doing their chores, or presenting a sugar pellet to a rat when it presses a lever are all examples of positive reinforcement. 2. Negative reinforcement also increases a behavior, in this case because it removes an aversive stimulus. Taking medication to relieve a headache or dressing warmly to avoid the cold are behaviors supported by negative reinforcement. Negative reinforcement can be tested in the lab by training animals to escape to another compartment when an aversive stimulus, such as a shock, is turned on. 3. Punishment decreases behavior through a contingency between a response and an aversive event. It includes slapping a dog for chewing slippers, scolding a child for making a mess, or fining a driver for parking in a reserved spot. In the lab, shocking an animal for lever pressing is an example of punishment. 4. Omission training, sometimes called negative punishment, decreases the probability of responding by withholding an outcome when the response occurs. Timeouts for young children work on an omission schedule. If the child behaves inappropriately, playtime and toys are removed for a short period. In the lab, not presenting a food pellet when a rat presses a lever is an example of omission training. In these cases, the animals are initially trained to lever press for food. Figure 4.2. The relationship between a response and its outcome in operant conditioning. An operant response may produce or prevent an outcome that is either aversive or appetitive, leading to 4 different reinforcement relationships. Okay, so the response can either prevent an outcome or produce an outcome, and the outcome can either be positive, appetitive, or negative, aversive. So the response is the X variable and the outcome is the Y variable. Okay, so for positive reinforcement, the response produces the outcome and the outcome is positive, appetitive. For negative reinforcement, the response prevents the outcome and the outcome is negative, aversive. For omission, the response prevents the outcome and the outcome is positive, appetitive. For punishment, the response produces the outcome and the outcome is negative, aversive. Experimental paradigms. Over the last century, over the last 100 years, most studies of classical and operant conditioning were conducted in the lab using rats or pigeons as subjects. This has changed in the last 2 or 3 decades as behavioral paradigms have been developed for other animals, including birds, fish, and invertebrates. Both forms of associative learning have been examined in the natural environment in a variety of species, although these are less common than lab-based studies. This is particularly true of operant conditioning, which is almost always studied in the lab. Classical conditioning. Most research into classical conditioning uses some variation of the paradigms described below. Conditioned preference. Many unconditioned stimuluses elicit positive emotional reactions, such as happiness or excitement, that can be conditioned to stimuli predicting their occurrence. Common examples are pleasurable sensations evoked by the scent of a sexual partner or feelings of comfort evoked by the smell of a familiar holiday meal. With few exceptions, organisms will approach stimuli that elicit positive emotional reactions, so classical conditioning is often manifested as approach responses to stimuli associated with rewards, such as food. This phenomenon was documented in carrion crows, which approached and explored mussel shells after a small piece of beef was placed under the shell. The dependent measure, approach behavior, was assessed by examining crow footprints in the sand, see figure 4.3. Initially, crows ignored the shells, but after they serendipitously and repeatedly uncovered the beef, birds began to head directly to the shells. Insects, including many species of bumblebees, adjust their foraging behavior in the same way, approaching flowers of a particular color or shape that have been associated with higher nectar content in the past. Figure 4.3, Crow footprints in the sand. In these field studies, crows and bumblebees developed conditioned approach responses to classically conditioned stimuli. The same phenomenon is measured in the lab using a conditioned place preference task, in which animals prefer to spend time in an environment where they experienced natural rewards, such as food, water, or sexual interactions. Non-natural rewards, including drugs and artificial sweeteners, produce the same effect. Importantly, conditioned preference tests are conducted in the absence of the unconditioned stimulus. Pavlov's dogs salivated when they heard a bell, even if food was no longer presented. Similarly, rats return to a place where they encountered a sexually receptive partner that is no longer present. In conditioned preference tests, the latency to approach the conditioned stimulus, the number of conditioned stimulus approaches within a given time period, and the time spent in close proximity to the conditioned stimulus are all indices of classical conditioning. Conditioned taste preference is a variation on this methodology, measuring increased consumption of a flavored food that was paired with a natural reward, e.g. sucrose. Using these paradigms, preference conditioning has been documented in a wide range of mammalian species, as well as invertebrates such as California sea snails, nematodes, and common fruit flies. Conditioned emotional reactions. Negative emotional reactions, such as anxiety or fear, may also be conditioned to stimuli predicting an unconditioned stimulus. Some people feel nervous or uneasy when they enter a dentist's office or an examination hall based on their previous experience in these environments. Animals display similar responses to stimuli associated with aversive events. For example, many emit alarm calls and/or escape responses when they encounter a predator. The sight, sound, or smell of these predators will, over time, elicit similar responses. In addition, if rodents or other small mammals mistakenly touch a sharp object or eat something that makes them ill, they respond to these stimuli by vigorously digging around the object and covering it with dirt or bedding. Importantly, this response occurs when the unconditioned stimulus is no longer present, i.e., they do not touch the object or taste the food, confirming that the conditioned stimulus is eliciting the conditioned response. In the lab, conditioned emotional reactions are commonly measured using the conditioned fear paradigm. These tests capitalize on the fact that frightened rodents often freeze, particularly when escape is unlikely. This may be an adaptive trait, as mice that freeze when they detect a predator before they are detected themselves are less likely to be attacked and killed. In the lab, freezing as a conditioned response is quantified by measuring the period of immobility, i.e., the conditioned response following the conditioned stimulus presentation. The conditioned suppression paradigm is a variation on this measure. First, rodents are trained to lever press for food. Once lever pressing is stable, classical conditioning trials are instituted in which a conditioned stimulus, usually a tone or light, is paired with a shock. When animals freeze, they stop lever pressing, so the number of lever presses that occur during the conditioned stimulus versus the number of lever presses that occur during non-conditioned stimulus periods is a measure of conditioned fear. This suppression ratio, as shown in figure 4.4, is typically calculated as lever presses during the conditioned stimulus divided by lever presses during the conditioned stimulus plus lever presses during an equal period of time preceding the conditioned stimulus. Figure 4.4, Conditioned Suppression Data. The conditioned suppression ratio is a common laboratory measure of classical conditioning in rodents. Lever pressing rates are recorded during the presentation of a conditioned stimulus, CS, such as a tone, that was previously paired with an aversive unconditioned stimulus, such as a shock. A suppression ratio of 0.5 indicates no change in responding when the conditioned stimulus is presented, thus no conditioning. With repeated conditioned stimulus, unconditioned stimulus pairings, the conditioned suppression ratio declines, indicating that lever pressing is reduced during the conditioned stimulus, creating a conditioned response. Okay, so we have a graph. On the x-axis is conditioned stimulus, unconditioned stimulus pairings from 0 to 6, and on the y-axis is the suppression ratio from 0 to 0.5. So you can see the more times the conditioned stimulus is paired with the unconditioned stimulus, the suppression ratio goes down from its beginning at 0.5 and it approaches 0 as conditioning increases the number of pairs of times. Thus, a suppression ratio of 0.5 would indicate no reduction in responding and no conditioning, whereas a suppression ratio of 0 would reflect complete freezing during the conditioned stimulus and maximal conditioning.